Hello, everyone. Thank you so much for joining our webinar today. I'm excited to have Kate and Lukasz here with me today, hosting the webinar. I am Amy, and I will be moderating today's session. Before we begin, I just wanted to let you know a little bit more about how the webinar will run. The webinar is being recorded, and the slides and recording will be available to you within 3 business days. A short Q&A session will follow the presentation to answer any viewer submitted questions. During the webinar, you can simply submit your questions using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions at any time. That concludes my very brief introduction today. Kate, I will now pass the webinar off to you. You may begin.
Thank you, Amy. And hello, everyone. My name is Kate Mostowska. I am a Chief Scientific Officer at Eurofins Food Division. I also have a global role within Eurofins, where I work with experts and business leaders in Eurofins pesticide residue testing labs on different topics and projects, including some of the topics which we'll be discussing today. And I also want to introduce my colleague, Lukáš Václavík. Lukáš, please.
Hello everybody, my name is Lukáš Václavík and I'm working as a technical manager at Eurofins Food Chemistry Testing in Madison, United States. I support contaminant and residue analysis. I work on developing and validating new methods for testing of residues, contaminants, and also adulterants.
Thank you, Lukáš, and we'll now move into the presentation. So today we'll be discussing determination of pesticide residues, heavy metals, and other important contaminants in dietary supplements and ingredients. But before, a little bit about Eurofins. From the global perspective, Eurofins is a large network of laboratories in almost 60 countries that provide testing services in many different areas, including food, environmental, pharmaceutical, or cosmetics testing, and also agroscience, genomic, forensic, and other services. This map shows the Eurofins US food footprint, with labs providing various testing, consulting, and also product design services. And as for today's topic, we have 2 labs specialized in chemical contaminant and residue analysis, one in New York and one in Madison, Wisconsin. And we have a long history of testing dietary supplements and ingredients for pesticide residues, heavy metals, and many other contaminants. I will start with some regulatory background. When it comes to regulations in the US, the essential regulation related to the supplements is the Dietary Supplement Health and Education Act of 1994. It amended the Food, Drug and Cosmetic Act, which is codified in Title 21, Chapter 9 of the United States Code. And this amendment, among many other things, provided definition of the supplements and ingredients. It highlighted that dietary supplements are deemed to be considered foods for regulatory purposes, and also that it is prohibited to market products that are adulterated and misbranded. And what that means is defined in Section 342, which gives many reasons for food to be deemed adulterated. And when it comes to chemical safety, there are included provisions for poisonous substances, pesticide residues, food additives, substitute added constituents, and also dietary supplement and ingredient safety. And also that dietary supplements have to follow current good manufacturing practices, otherwise they are deemed adulterated. And the current and good manufacturing practice in manufacturing, packaging, labeling, or holding operations for dietary supplements is given in 21 CFR Part 111, which, among other important things, requires to establish specifications for each component used in dietary supplement manufacture for their identity, purity, strength, composition, and limits on contamination that may adulterate or may lead to adulteration of the finished batch of the dietary supplement. And then, again, among many other things, there is a requirement to verify that the finished batch of the dietary supplement meets product specifications via tests and examinations. So testing is definitely very important. When it comes to chemical residues, contaminants, and adulterants, here's a list of different compound groups be typically considered in risk assessment of dietary supplements when designing testing programs for ingredients and finished products. And today, Lukáš and I will discuss the most important ones that are of concern in the majority of sample types, especially those of botanical origin. Those include pesticide residues, heavy metals, mycotoxins, and residual solvents. And then also cover pyrrolizidine alkaloids and pharmaceutical adulterants that are of concern in certain ingredients and products. And I will start with pesticides and residuals and then turn over to Lukáš for the rest of the groups. When it comes to pesticides, there is sometimes a confusion what a pesticide actually is, because pesticides include a wide range of compounds with different biological activities and use. Primarily, pesticides are intended for preventing, destroying, and controlling pests. And from that perspective, the main pesticide classes include insecticides to control insects, fungicides to control fungi or mold, and for weeds. However, there are also compounds such as plant growth regulators, defoliants, desiccants, or agents for thinning fruit or preventing the premature fall of fruit, which are also included in the definition of pesticides. And also, are substances applied to crops either before or after harvest to protect the commodity during storage and transport. An example of a substance that is used for harvest is the fumigant ethylene oxide, which I will talk about in more detail in a moment. As I mentioned, there are many different groups of compounds with different biological activities within the pesticide, and there are also diverse. They also have diverse structures and chemical properties. There are well over 1,000 different pesticides, and their structures are really quite different. I just included a few examples here. You can see very small molecules, very small, basically things like bromide ion, all the way to, for example, an example here of abamectin on the left. When it comes to polarity, we have compounds which are fairly non-polar, like DDT, for example, all the way to, for example, glyphosate. So very different properties and structures. So obviously, there are just very few examples here because the number of compounds is quite large, which together with the diversity of the compounds pose a challenge for pesticide residue analysis. In addition to the low target concentrations and also to the complexity to matrices, which is certainly the case for dietary supplements and ingredients. The current best practice is to analyze the majority of important pesticide residues using so-called multi-class, multi-residue methods, which typically use a combination of gas chromatography with tandem mass spectrometry, or GCMS-MS, and chromatography combined with tandem mass spectrometry, or LCMS-MS, for the determination of a wide range of compounds. Here's an example from our US labs for the analysis of over 500 pesticide residues, where we have a significant overlap between the two analytical plots, meaning that certain compounds are analyzed by both GCMS-MS and LCMS-MS, which provides increased confidence in identification of potential hits. Another important thing to mention is that we spend a lot of time optimizing the MS-MS transitions, just for sensitivity, but also, or really highly importantly, for high selectivity, which is crucial in complex dietary supplement products and ingredient matrices. In addition to multi-class, multi-residue US, we also offer various single residue or single class methods, because some compounds require special analytical preparation, such as hydrolysis or conversion step, or special chromatographic conditions. Some of the examples include diethylates, inorganic bromide, already mentioned ethylene oxide, or pesticides such as glyphosate. We also offer testing packages to meet specific requirements, such as the USDA National Organic Program, or NOP, list of pesticide residues, or evaluation of compliance to the NSF ANSI 173 standard, which includes a list of pesticides based on the National Organic Program NOP list, together with daily maximum allowable levels, which we use in the evaluation of the determined residues. And last but certainly not least is the United States Pharmacopoeia, USP, the General Chapter 561 for articles of botanical origin, which lists selected priority pesticides and with limits. And for botanical extracts, the limits could be adjusted based on the concentration factor and equations which are provided in the USP Chapter 565. And here is the here are the pesticides listed in the USP 561 together limits in milligram per kilogram. I'm sorry for a small font, this is just for illustration. Okay, so you can find the details in the actual chapter. These limits are pesticide residue dependent, ranging from 0.01 milligram per kilogram or 10 ppb for compounds like Myrex or pentachloronazole to 125 ppm, 125 milligram per kilogram for inorganic bromide. And we analyzed most of these compounds, the majority of the pesticides, pesticide residues listed in the USP 61 using a multi-class, multi-residue method based on the combination of GCMS-MS and LCMS-MS. And also want to highlight that we use the method of standard addition for accurate quantification of residues which are close to the USP limit. In this way, we compensate for any potential matrix effects. In case of inorganic bromide, our methodology is based on the EU reference laboratory method, which involves derivatization of inorganic bromide with propylene oxide. For dietary carbamates, we employ a method that uses LCMS-MS for the analysis of methylated analytes with preserved dietary carbamate moiety. And this approach is more selective than the traditional conversion of dietary carbamates to carbon disulfide, or CS2, which is developed by the labs. And this approach can lead to potentially false positives, because carbon disulfide could be also generated from natural sulfur containing compounds which are present in sample matrix. So now, as I promised, I will talk a little bit about ethylene oxide, because there's been a growing concern related to this compound, which started with residue findings in sesame seeds coming from India to the EU, where ethylene oxide is banned as vegan. But later, this concern expanded to other commodities and products, including dietary supplements and ingredients. So what is ethylene oxide and why are we concerned about it? Under normal conditions, ethylene oxide is a gas. It has no color and it's flammable. It is the smallest epoxide. It is really quite small molecule, as you can see here, and it's highly reactive, which makes it useful in many applications, but it is also a reason behind its toxicity, because it's been classified as carcinogen compound. I listed some of the other concerns, mutagenicity, and also toxicity to reproduction here by different organizations. When it comes to the use of ethylene oxide as food fumigant, how does it work? that, as I mentioned, ethylene oxide is highly reactive. It is a strong alkylation agent, and it reacts with various organic compounds, including enzymes and other proteins or DNA. And this way, it denatures important functional proteins and enzymes and causes extensive damage to the DNA. And as a result, this alters cell metabolism and inhibits growth and reproduction, so it can control bacteria, including spores, fungi, and viruses. And as a very small molecule, it has high diffusivity and strong penetration, which makes it very effective in dry food commodities. And so it's mainly used on spices, nuts, oil seeds, dried fruits and vegetables, and also dried herbs and botanicals. When ethylene oxide is applied on food, it dissipates through evaporation and various reactions with food matrix. And in addition to the reactions with organic compounds, which I discussed previously when it comes to reactions with proteins and so on, it also reacts with chloride ions to form chloroethanol. And this molecule, which is shown here, 2-chloroethanol, is less volatile and also less reactive than ethylene oxide. So its removal from the samples is limited. And so so this compounds, 2-chloroethanol, serves as a marker residue for ethylene oxide use for regulatory compliance purposes. And as I mentioned, ethylene oxide is regulated as a pesticide. It's approved in the US. It's approved for uses on several commodities, which are listed here, including herbs and spices, and also, for example, dried vegetables, among other things. And this table shows the US tolerances, which means maximum residue levels allowed on these commodities for ethylene oxide and 2-chloroethanol, which, as you can see, are very high ppm or milligram levels. In the EU, the situation is different. As I mentioned, in the EU, ethylene oxide is not approved as food fumigant. It's used for this application has been banned since 1991. And as such, maximum residual levels, or MRLs, have been set at a limit of quantification, or LOQ, reasonably achievable in different commodities. And they are set for the sum of ethylene oxide and 2-chloroethanol expressed as ethylene oxide, which is the definition for ethylene oxide in the EU. For food supplements, the EU Commission stated that for harmonization and consistency purposes, the applicable MRL is 0.1 milligram per kilogram, including capsules used for production of food supplements. So that's important information which was added later on when the dietary supplements and ingredient became of concern and had some findings of residues. When it comes to the analysis of ethylene oxide and 2-chloroethanol, US labs adopted a method developed by our Eurofins colleagues in Germany, who were at the forefront of the testing from the beginning of the ethylene oxide crisis in Europe. This method involves conversion of ethylene oxide to 2-iodoethanol and analysis of 2-iodoethanol and 2-chloroethanol using GC-MS-MS. And this approach provides robust routine analysis in a wide range of food matrices and enables separate reporting of ethylene oxide and 2-chloroethanol, as well as reporting as the sum down to 0.0 milligram per kilogram. And this method sharing and other collaboration within our global Eurofins network has been really useful for managing the ethylene oxide crisis and supporting clients in different regions. Because the last time I checked, we had 13 labs in 8 countries testing ethylene oxide and 2-chloroethanol residues in foods, including labs in Asia, Europe, and the US. And now just briefly about residual solvents. The USP chapter 67 defines acceptable amounts of residual solvents in pharmaceutical drugs and dietary supplement products, and also includes a definition of residual solvents, which are organic volatile compounds. So from analytical perspective, the choice is headspace, GC analysis, and for us combined with mass spec detection. The USP 467 provides classification of residual solvents based on their risk assessment into class 1, which are solvents to be avoided, class 2, solvents to be limited, and class 3, solvents with low toxic potential. And for each of these classes, the chapter provides a list of solvents with limits, which are used for specifications as specifications for testing. So that was just briefly residual solvents. And now with that, I will now turn it over to Lukáš, who will discuss the other important contaminants. So Lukáš, I will stop sharing and
all right, thank you so much for that presentation, Kate. And now we'll move into Lukáš. I just gave you the presentation, so I'll pass it off to you.
Thank you very much. Okay, let's move forward with this presentation, starting with an important contaminant group, which are heavy metals. When it comes to heavy metals, arsenic, cadmium, lead, and mercury are typically of highest concern due to their toxicity. These and other heavy metals are taken up from contaminated soil by plants during cultivation, and when used as botanical ingredients, metals are transferred to finished products, which, depending on the processes used, may be accompanied by concentration of their levels. Soil contamination usually comes from industrial sources, irrigation, or use of poor quality fertilizers and plant protection products. Botanical materials may also be contaminated during processing, during milling and other processing steps that are being taken to prepare finished product. Many dietary supplement producers use the US Pharmacopoeia limits for heavy metals in finished products, which are provided in chapter 20 to 32 as specifications. So they use these limit as specifications. These are based on permitted daily exposure values, and in order for dietary supplement finished dosage form to comply with the limits for these environmental contaminants, the level of elemental contaminants in that dietary supplement should not be more than the permitted daily exposure. And there are multiple options for determining the compliance with these limits. The typical heavy metal analysis workflow includes thermal decomposition of the organic components of the sample with the use of inorganic acids and other reagents. This process is called mineralization, and this is followed by a dissolution of the residues in suitable solvent and analysis by ICP-MS technique, which stands for inductively coupled plasma mass spectrometry. This approach allows determination of total concentrations of individual heavy metals down to low ppb levels, even in highly complex samples, including botanical dietary supplements and ingredients. In addition to total heavy metal analysis, determination of respective species of metals is of high importance because there may be significant differences in their toxicity. The inorganic species of arsenic, which are arsenite and arsenate, are highly toxic, while organic species are not considered toxic. So we do have a method available that allows determination of both toxic inorganic species along with common arsenic organic species, and we are able to achieve typical limits of quantification around 10 ppb for each individual species included in this method. The way this is achieved, we basically include chromatographic separation using ion chromatography to separate respective arsenic species, and these are eluted out of the column and detected by ICP-MS technique. Another speciation assay I would like to highlight, and we are currently still working on this assay, is method for chromium speciation, where the goal is to determine side-by-side trivalent and hexavalent chromium that again differ in their toxicity. This represents a rather big challenge because the species do convert to each other depending on the conditions. This takes place during sample preparation, means extraction, and also during instrumental analysis. So depending on the pH and matrix components, it can be really difficult to make sure that we are determining real concentrations in samples, and we do not see interconversion. So this is work in progress, something that will be available in near future. Okay, talking about mycotoxins, these represent a diverse group of naturally formed chemicals that are of high concern with regard to their toxicity. These toxins represent secondary metabolites of species of fungi, and of course contamination with mycotoxins can take place both during cultivation and storage of plant materials. USP 561 provides limits for aflatoxins in articles of botanical origin. It should be noted that these USP limits are aligned with action levels for aflatoxins in foods issued by the US FDA administration. In the European Union, another set of regulatory limits for a wider range of mycotoxins is provided in Commission regulation. You can see this slide. Considering relatively low target concentrations, we need effective sample cleanup for analysis of mycotoxins, especially when it comes to complex sample matrices. The workflow implemented in our laboratory is based on immunoaffinity sample cleanup, which is followed by liquid chromatography with tandem mass spectrometry determination. In our workflow, the analytes are extracted from the sample using a combination of suitable solvents, and the resulting extract is diluted and loaded on the immunoaffinity column. Column contains antibodies that are specific to mycotoxins of interest, and while the analytes are bound, we are able to flush the sample matrix components out of the column and finally elute the analytes, concentrate them, and analyze by LCMS. In this slide, we do see a summary of the workflow we use. As you can see, we can use a single column to perform cleanup for all these regulated mycotoxins, and we are also able to achieve compliant reporting limits or limits of quantification in complex samples. Okay, another group of important contaminants that gained lately a lot of attention are pyrrolizidine alkaloids. There are over 500 different PAs and their N-oxides known to date, and these are basically secondary metabolites that are produced by various plant species as a defense chemicals against herbivores. These compounds were shown to be hepatotoxic and genotoxic, and their toxicity may dramatically differ between various subclasses, and as usual, is associated with the structure of these compounds. Contamination of supplements and ingredients is typically associated with co-harvesting of alkaloid-containing weeds or accidental inclusion of such plant materials in herbal ingredients. Alternatively, cross-contamination of crops that do not produce pyrrolizidine alkaloids in the field can occur via uptake of these toxins from soil, and the soil is basically contaminated from rotting and living donor plants that contain pyrrolizidine alkaloids. Reduction or elimination of these compounds in ingredients and finished products is really challenging task that requires complex processes and integration of various mitigation measures at both cultivation and harvesting stage. The issue of pyrrolizidine alkaloids contamination of supplements has been recognized by the US Pharmacopoeia, and currently there's a draft general chapter available that summarizes information on structure of pyrrolizidine alkaloids, their stability, dietary exposure, toxicity. Also list the most important pyrrolizidine alkaloids that are abundant and should be monitored in various matrices, and also provides a summary of tolerable daily intake levels. And last and not least, there's a discussion or information on suitable analytical methods to test for these compounds and also associated challenges. On July 1st, 2022, a new European Union regulation became effective that sets the maximum limits for lower bound sum of 21 pyrrolizidine alkaloids and their N oxides. That these also include additional compounds that may currently analytically interfere. That means collude with target analytes, those 21 compounds basically. When it comes to regulated matrices, these are herbal infusions, teas, dietary supplements, pollen-based supplements, herbs, and spices. In our lab, we worked on a method. We are using UHPLC coupled to tandem mass spectrometry. We use SPE cleanup for sample purification, and we also employed isotope labeled internal standards to help us to get more accurate results. The method was thoroughly validated, and we were able to achieve reporting limits that are in agreement with regulatory levels in the European Union. So this method we have is suitable for testing of dietary supplements and other matrices, to comply with the new regulation. Moving forward to another group of contaminants, or rather adulterants, which are pharmaceuticals. So basically, adulteration of dietary supplements with pharmaceutical ingredients is a practice that is profit-driven. The main goal is to develop a biological effect that is basically for which the dietary supplement is advertised, for example, weight loss, etc. So consumers are thinking they are consuming a all natural product, but the product is actually spiked with pharmaceutical adulterants. The pharmaceutical ingredients that are used for adulteration are maybe various approved drugs, but also withdrawn drugs, or even drugs that haven't been ever tested, just didn't make it into any type of clinical studies. So these designer analogs are, we have no information on their safety profile. Additionally, and this adds to the severity of this problem, dietary supplements are typically adulterated at high concentration, and on occasions with multiple compounds, multiple pharmaceuticals. Those high concentrations are used to develop the effect, so they need to be close to therapeutic doses. And when it comes to most critical supplements, these are supplements for bodybuilders and sport supplements, weight loss supplements, and also male sexual enhancement supplements, which are being added with different pharmaceuticals depending on the claimed effect. So obviously, anabolic steroids or selective androgen SARMs, basically, which stand for selective androgen receptor modulators. Weight loss would be spiked with anorectic drugs, diuretics, or laxatives, and sexual enhancement would be spiked with PDE5 inhibitors compounds such as tadalafil, sildenafil, etc. This problem is being recognized by the US FDA, and they are looking into it in detail, testing dietary supplements sold over the counter and also online. And there was a number of warning letters that were sent to small businesses, but also big players, noticing or basically highlighting this issue and requesting some kind of remediation. So with this problem, Amazon, which everybody knows, it's a huge e-commerce portal. They came up with a policy for dietary supplement sellers. They basically need to demonstrate, their product is of sufficient quality, and they need to provide evidence to verify the label. But on top of that, for certain high-risk supplement categories, which I just outlined on the previous slide, they require testing for selected adulterants, which are being selected based on frequency of detections, I think, to a great extent based on contents of warning letters by the FDA. So we have PDE5 inhibitors for male sexual enhancement drugs, anorectics such as sibutramine or its analog desmethylsibutramine, laxative phenolphthalein in weight loss management, and then we have SARMs, ostarine ligandrol, in sports nutrition, along with 4-DHEA, which is an anabolic steroid, and again some weight loss drugs and stimulant DMMA. And Amazon basically requires to perform this testing annually. In our lab, to serve our customers, we developed 3 separate methods that are based on LC-MS, and they are basically their scope matches the Amazon requirements. This is just an overview of the reporting limits we are achieving. These differ depending on the actual therapeutic doses needed to generate biological effect. And these methods, allow us to screen for the presence, and if found, we can also quantify the pharmaceutical adulterants in these supplements. On top of these rather limited methods that cover rather limited number of analytes, we also have larger scale screens for PDE5 inhibitors. In total, we are able to monitor 50 PDE5 inhibitors and their structural analogs. And we have separate method also for weight loss agents and stimulants. And we are using liquid chromatography coupled to high resolution mass spectrometry to screen for these compounds. We are also able to use this technique to quantify respective levels. And considering this instrumentation is high resolution mass spectrometer, we are not looking only at targeted compounds for which we have standards, but we are also able to look for novel analogs which typically share the backbone of the molecule, but are modified, slightly, as is documented for analogs of sildenafil in this slide. So specifically method for PDE5 inhibitors, we did perform validation and submit this method to address call for screening and quantification methods to analyze PDE5 inhibitors in dietary supplements, and our method became official method of analysis AOAC International. So just to document what we are doing to look for analogs and how we are detecting or collecting data, we perform analyses in full scan mode, which basically covers all the masses. We are able to monitor both targeted and also untargeted analytes by looking for respective molecular ions. For targeted compounds, we collect data-dependent MS-MS experiments to collect product ion spectra for these targeted compounds, which allow us to identify these analytes with high confidence. And on top of that, we also run all ion fragmentation experiment, which allow us to generate fragmentation spectra for additional compounds we are not aware of, which may be in the sample as well. And then we have smart or advanced data processing tools to look for these analytes or compounds that share certain characteristic signals in their fragmentation spectrum, and we are able to highlight suspect peaks that may represent a novel analog of different drugs, as demonstrated in this slide. So using this approach, we are basically able to highlight unknown adulterants that may be present in your sample, and moving forward, using this technique, we are able to assign probable structure and attempt identification of that compound. That was my last slide. I would like to acknowledge our colleagues at Eurofins Central Analytical Laboratories in New Orleans and also Eurofins Food Chemistry Testing in Madison, Wisconsin for all their contributions to this presentation. And of course, I would like to thank you for your attention. Kate and I, we are looking forward to your questions in QA session.
Wonderful. Thank you so much, Lukasz. So just as a reminder to everyone, please continue to submit your questions in the Q&A section. We will review these, and if we do not have time to address them today during the live presentation, we will follow up with you directly after the webinar. So it looks like we have several questions, though, that have come in. The first one, I will kick it off here. Is there an actual method for pesticide residue analysis included in the USP Chapter 561?
I guess this one is for me. So no, there is no method in the current USP 561. But in addition to the list of pesticide residues and their limits that I showed in one of my slides, there are requirements provided in the chapter in terms of, for example, recoveries or RSDs. But actually, years ago, there was a very outdated method included in the chapter in the USP 561 that could not really analyze the listed compounds. And back then, I proposed revisions to the USP, which were mostly adopted, so that method was removed. And actually, recently, I saw some debate about the use of description as USP 561 modified as a method reference, which I think might have been used by the labs that have been testing the USP 561 list of compounds for a long time and simply didn't update their reference description because, again, that method was removed from the chapter some time ago.
Great, thank you. The next question here, do you have testing capabilities beyond the mycotoxin scope you showed in your presentation?
I'll take this one.
Okay.
Sorry. Yeah, the simple answer is yes, we do. In addition to what I've shown in the presentation, we do have additional multi-method that covers over 40 mycotoxins and includes also their metabolites and some alkaloids. You know, there's a lot of compounds, but for example, we include citrinine or eniantines in this method. And last but not least, we are also able to test for ergot alkaloids. This method has been used mainly for study work, but it's fully applicable to dietary supplements as well. Just to note, in this case, we are not using immunoaffinity column cleanup because we don't have columns that would accommodate 40 plus mycotoxins, but the method works, and we are able to monitor those compounds and quantify as well.
Wonderful, thank you. Next question here, let me pull this up. Are there any AOAC official methods being developed for contaminants or adulterants in dietary supplements and ingredients?
Yes. Actually, Lukáš mentioned the official method for PDE5 inhibitors, which we developed to address the industry need several years ago, because PDE5 inhibitors were identified among the top high priority issues by the AOEC panel on dietary supplements. Back then, this program was supported by NIH Office of Dietary Supplements, and this was a program which was a 5-year program and ended in 2018. However, and within that program, there were multiple methods developed into official methods. However, AOEC has just initiated a new program on botanical and dietary supplement integrity, and Eurofins is one of the founding organizations. And one of the seed projects is determination of pyrrolizidine alkaloids that Lukáš discussed these compounds in his presentation. And I believe that there will be other projects focused on contaminants in this new AOEC program. And on top of that, there is also the AOEC cannabis analysis or cannabis analytical science program, or CASP, which covers residues and contaminants in cannabis, including hemp and hemp-derived products. So that's in a nutshell kind of an overview of what's happening at AOEC related to contaminants in supplements.
Wonderful. Thank you, Kate. And I think we have time for one more question here. Considering the complexity of dietary supplements and botanical ingredients, how do you cope with quantification? There may be strong matrix effects that affect analyte signals, and quantification with solvent standards may not always be available or possible.
Yeah, I totally agree. Matrix effects we observe are in some cases really extreme, and due to large differences in respective test samples we obtain for testing, we cannot really use matrix matched calibration. It's possible in certain cases, but not always. Which leaves us with several additional options. We can try to dilute out the matrix effects, which typically affects the LOQ and reporting limit. The other option is the use of isotope labeled internal standards that typically allow for effective compensation of matrix effects, and we are trying to include those in our methods as frequently as possible. And another approach we typically take for ensuring accurate quantification is the standard addition method, which allows compensation for matrix effects, but also compensates for any analyte losses during the extraction or instrumental analysis. So this is really like the best option we use, and we employ it when we really need highly accurate quantification.
Wonderful, thank you. And it looks like we're coming up to the end here, so I think that will wrap up our Q&A session. I want to remind everyone that a recording of the webinar, along with a copy of the slides, will be available within 3 business days. Thank you again, Kate and Lukasz, for your presentation, and thanks everyone for attending today.
Thank you, Amy. Thank you, everyone.
Thank you.